Good morning, everyone. I am Prakhar Dikshit from Chemistry Honors, and today I will be presenting on the structure of polynucleotides. We will explore DNA, RNA, and the forces that help maintain their structures. Basically, these molecules are incredibly important because they hold the instructions for life. Every trait, whether it's your eye, color, height, or metabolism, is ultimately controlled by DNA and expressed through RNA. So, understanding their structure helps us understand how life actually works at the molecular level. So, now move to the next slide. So my second slide shows what is a polynucleotide. Basically, a polynucleotide is a long chain made of repeating nucleotide units, and each nucleotide has three components: a sugar, a phosphate group, and a nitrogenous base. These nucleotides are linked by 3,5-phosphodiester bonds, which form the backbone. And this backbone is like a structural framework that holds the entire molecule together. Even though DNA and RNA are both polynucleotides, small differences in their sugar and bases completely change their function. For example, DNA's deoxyribose makes it chemically stable, while RNA's dose makes it more reactive and able to fold into many shapes. These differences explain why DNA is ideal for long-term information storage, while RNA is perfect for short-term tasks and catalytic roles. Let's move on to the next slide. This slide shows the DNA structure. Basically, DNA is one of the most beautiful molecules in biology. Its double helix structure resembles a twisted ladder. The two strands run in opposite directions, called anti-parallel orientation. This arrangement is important for enzymes during replication and transcription. Base pairing follows specific rules, like A pairs with T using two hydrogen bonds and G pairs with C using three hydrogen bonds. GC pairs have an extra hydrogen bond and stronger stacking interaction. Regions rich in G and C are more thermally stable. This is why organisms living in extreme environments often have GC-rich DNA. The helix turns after approximately 10.5 base pairs, giving DNA its characteristic spiral shape. This structure is compact yet stable enough to store huge amounts of genetic information in every cell. The slide 4 shows major and minor groups. When the DNA double helix twists, it creates two groups, the major and the minor groups. These groups are not just structural, they serve as recognition sites. Proteins that regulate genes, repair DNA, or replicate it often bind in the major group because it's wider and offers easier access to the base sequence. The minor group is narrower but still important for certain types of proteins and drug binding. These groups basically act like a molecular addresses that help proteins find the correct spot on the DNA quickly and accurately. Slide 5 shows RNA structure single-stranded versatility. Unlike DNA, RNA uses ribose sugar, which has a reactive 2OH group. It also contains uracil instead of thymine. RNA is usually single-stranded, but this is what gives it incredible flexibility. It can fold into hairpins, loops, bulks, and many complex shapes. Because of this structural diversity, RNA can do much more than DNA. It can act as a messenger, an adapter like tRNA, or even an enzyme as seen in ribozymes. When RNA forms double-stranded regions, it always takes on the A form helix, which is shorter and wider than DNA's B form. This compact structure helps RNA maintain stability despite being single-stranded. To the next slide. Slide 6 shows forces stabilizing DNA. One reason DNA is so stable is because several different interactions work together. First, hydrogen bonds provide specificity in base pairing. Second, base stacking creates van der Waals forces between neighboring bases, giving the helix strong internal stability. In fact, base stacking contributes more to DNA stability than hydrogen bond. Third one is hydrophobic interaction, push the nitrogenous bases inwards, away from water, while the sugar phosphate backbone faces outwards. Finally, ionic interaction with metal ions like Mg2 plus and Na plus helps neutralize the negative charges on the phosphate groups, reducing repulsion. These are the additional DNA stabilizing factors. There are two more interesting stabilizing factors. One is DNA length and second is supercoiling. Longer DNA strands naturally form more hydrogen bonds and have more stacking interactions, making them more stable. The second is supercoiling. Some enzymes introduce twists into DNA to either tighten or loosen it. Negative supercoiling, which is common in cells, helps reduce torsional strain and makes DNA more compact, easier to unwind during replication. These are the forces that stabilizes the RNA. RNA stability is slightly different because of its single-stranded nature. Its stability comes from regular base pairing like AU and GC, non-stranded pairs like GU wobble, strong base stacking in helical regions, hydrogen bond networks in loops and junction. These features helps RNA maintain its shape even though it's chemically more reactive than DNA. These are the advanced RNA stabilization factor. More complex RNAs such as tRNA and RNA rely on additional stabilizing mechanisms. Mg2 plus ions are especially important. They bind tightly to the phosphate backbone and are essential for proper folding. RNA also forms tertiary structures like coaxial stacks, which gives the molecule a compact 3D shape. This allows RNA to function not just as a messenger but also as a catalyst, a regulator, and a structural component of ribosomes. Because of these properties, many scientists believe that early life on Earth may have been based on RNA rather than DNA. And to summarize everything, DNA and RNA are both chains of nucleotides linked by phosphodiester bonds, and DNA forms a stable double helix, ideal for long-term information storage. RNA is flexible and folds into many shapes, allowing it to perform a wide variety of functions. Their stability comes from hydrogen bonding, base stacking, hydrophobic effects, ionic interaction, and complex tertiary folding. Together, DNA and RNA form the central framework of life. So that's it from my side. Thank you for your attention. This was my presentation on the structure of polynucleotides. I would be happy to answer any question. Thank you.